chapter 13, my main reason for chapter 13 is really the connection with the medical aspects of things, which I think are going to be interesting. Um, Basically, what chapter 13 talks about is diversity, right? It talks about all the different things that exist from a very simple single-celled bacterium, which is a prokaryotic thing, to a multicellular organism, which is what we are. Um, And so one of the things that's amazing about these single-celled organisms is how simple they are, how small they are, um, and how fast they reproduce. So the thing they have going for them is that they're not very complicated. They just got to get this right once. And then if they manage to make a second one of the same thing and it survives too, hey, more power to you. They've been around, single-celled organisms, prokaryotes, have been around for millions and millions of years. And they've spent millions of years exploring different means of energy sources. um, so the cyanobacteria, for example, in Hot Springs National Park, um, those are green, so they do photosynthesis. But there are other organisms that have different colors. That some of them feed off of arsenic, that is poisonous. It's amazing what bacteria can use as a resource. Um, Even like at hydrothermal vents, we thought that was like pretty impossible for anything to live there. Yeah, well, wrong. Um, Stromatolites, those are 1.5 million billion years old. Stromatolites are literally layers of dead microorganisms. So that's what stromatolites are. Um, Prokaryotes, we just basically distinguish them by their shape. They're small enough that you can see them under the microscope, but barely. So those of you who have, if you have 113 in any shape or form, you look under a microscope, you basically just see round things or long things or squiggly things. That's about the extent of it. And so that's how we distinguish them. We call them things like micrococcus something or another, right? Coccus meaning it's a round thing, or staphylococcus. You've all heard of staphylococcus before. Staphylococcus means that it's a circle, it's a coccus, and staphylo means that it's in a cluster. Staphylococcus, if you look at it on a under a microscope, you'll see it as like little round clusters. Um, so a typical bacterium kind of looks like this. It looks just like a simple cell. It doesn't have a nucleus. It's a prokaryote, so doesn't have a nucleus. Has a simple nuclear DNA. The thing that to us is the most interesting about the bacterium is the thing how they're different. The main thing how they're different from us is, inside of there the ribosomes are different. And then outside, the bacterial capsule and the cell wall are different. The cell membrane is actually the same as we do. That's all. That's all the same plasma membrane as we have. But the cell wall and the capsule are different from the way we are built. Um, and we use that to fight bacteria. So when you're looking at two different types, there's two different types of bacteria. We call them gram-positive and gram-negative bacteria. Mr. Graham discovered at some point that if you took bacteria and you dyed them with a purple dye, um, then that purple would stain all the bacteria. And then if you wash that purple dye out, it comes out only on some bacteria, not all of them. The ones where it doesn't come out, those have a really thick peptidoglycan layer. This thick peptidoglycan layer keeps you from being able to wash out this stain with alcohol. If you have a thin peptidoglycan layer, then this washes out like nobody's business. And so what people tend to do is they tend to have so this purple then gets counterstained with a red stain, and then the gram-negative bacteria tend to be pink, and the gram-positive bacteria tend to be purple. Um, the reason why this matters is because by doing this stain, you're actually distinguishing cells that are bacteria that are sensitive to penicillin and bacteria that are not sensitive to penicillin. It turns out that penicillin interferes with the production of the peptidoglycan layer. So anything that's gram-positive that needs a big peptidoglycan layer, if you give penicillin, it can't do it. It kills it. If your bacterium is gram-negative and it's got an itty-bitty little peptidoglycan layer, and you give it penicillin, it just laughs at you because it doesn't care. I mean, if one layer and no layer makes no difference really. So gram-positive bacteria, your those are the ones that you can fight with penicillin. This is why this matters. This is why knowing what bacteria are, what the bacterial structures are, this is why it matters. Um, Now, this here happens to be a gram-positive organism, Staphylococcus aureus. See, they're little cocci, little round things. Now, this little nasty little thing is methicillin resistant, so it's an antibiotic resistant. I don't remember how methicillin works, but it's another antibiotic that works in some way. Actually, it may be a penicillin. The fact that it's got the cillin, it probably is a penicillin derivative that normally would work on this, but it doesn't anymore. The way bacteria get resistant to antibiotics is actually pretty magical. What they do is they end up with little intracellular extra pieces of like extra chromosomal pieces of DNA called plasmids. Plasmids, Plasmid, oops, MID, and little plasmids, they carry that on the inside, and then they can pass it on. They can pass the resistance on to other organisms, to other prokaryotes that don't even have to necessarily be the same species. Um, so if one of your bacteria in your body gets penicillin resistant, all of them do. So that's why you don't want to get penicillin resistant. Um, there was an outbreak at some point of an E. coli outbreak. E. coli are sometimes harmless. Some of them are not so harmless. They cause diarrhea and can actually kill you. And in this particular case, it was it was with I think it was bean sprouts, right? It was locally grown bean sprouts that were contaminated with E. coli, that sickened a whole bunch of people. I remember when this happened. It was a big deal trying to figure out where this came from. We had one like that in the U.S. one time where lettuce was contaminated with something, and I don't think they ever quite figured out where it was, and you couldn't wash it off because it was internal to the lettuce, so that was pretty nasty. Um, Now, obviously, we use our prokaryotes to do stuff, right? Our yeast is very important to us, right? Molds for cheese, right? Soy sauce, that's also a yeast kind of thing. Cheeses are made with by little bacteria, prokaryote, lactobacilli, do that kind of thing. So we absolutely use our prokaryotes lots of different places. We bake bread, right? Yeasts do that. The other thing bacteria are capable of doing, which is pretty amazing, bacteria are able to. There are bacteria that can do just about anything out there. And so one of the things we actually use bacteria to is for bioremediation, for them to like bacteria literally can help us clean up oil spills. Um, Now this happens to be a mitochondrium, which if you've been to my spotlight class, I just recently talked about mitochondria. Mitochondria are in our cells, they're inside of our cells. But mitochondria actually were at some point in their life, they were individual prokaryotic organisms that literally gave up their lives to live with us in a symbiotic relationship, and we very much get a lot out of our mitochondria because they break down the energy for us. One of the things that's amazing about mitochondria, while they live inside of us, we don't encode for them. So you literally you don't have the genetic material like in your cells is the blueprint for everything in your body except for your mitochondria. You don't have a blueprint for those. You got those ready formed from your mother. So mitochondria only come from other mitochondria. Um, that's mitochondria for you. This is why mitochondrial diseases are really weird, because they're inherited differently, because they just going from mother to child and from mother to all the children because they just come from the mother's egg. So the endosymbiont theory, the way it goes, is and like I said, there's lots of evidence that speaks towards that idea. But the idea is that there was at some point there was a very early prokaryote, and that it became a cell that had things. And then at some point, it sucked up a bacterium that has the ability to do aerobic respiration of some sort. And it turned out that this combination with the mitochondrium and the cell was much, much more successful than without. So as a matter of fact, I don't know that there are any eukaryotic cells that exist out there that don't have mitochondria in them. Now, in a secondary event, the idea is that these cells that had the mitochondria in them already picked up another hitchhiker, which was a photosynthetic bacterium. And then this photosynthetic bacterium, those are the modern plants, basically, and those are the modern animals, basically. So that's the idea behind the endosymbiont theory. Um, Now, protists are your single-celled, microscopic kind of organisms. They can be, they can be anything from like a middle little single-cell thing, all the way to these kelps, which are entire underwater forests. Um, the way these little organisms eat, they do this thing called phagocytosis, and this is probably exactly how the first organism was. The first mitochondrium, the first chloroplast was engulfed. It just didn't get themselves digested. So a food particle kind of gets in. There's this invagination of the membrane that happens, and it just gets sucked up inside, then merges with a vesicle that has the digestive enzymes on the inside. And you got to keep track of digestive enzymes. You got to be careful with those because digestive enzymes will digest you just as much as they will digest your food. Um, and then the digested materials either gets excreted, or it may be getting reutilized for other things because everything gets recycled, right? Um, This is just the this idea of where protists are coming from and where they're going to. Um, this is red blood cells that are infected um, with P. falciparum. So everything that that we have can also be infected with things. So there are little bugs that are constantly fighting us. So these are trypanosomes um, amongst red blood cells. So that's bad news, right? Um, Plants are also being affected by both by both protists, and then later on with fungi and stuff like that, right? Um, there's also lots of symbiotic relationships, similar to the symbiotic relationships that we have with our mitochondria. So corals, for example, they have a symbiotic relationship with dinoflagellates. Um, and then we've got a whole completely different family of organisms, which is the mushrooms. The mushrooms are probably a whole lot more widespread than we give them credit for. They are in symbiotic relationships with probably more things than we give them credit for. We know we've known for a long time then there that fungi have a symbiotic relationship with trees. But it seems like basically every place we go, there's a whole entire fungal network underneath of our feet that moves nutrients from one place to another, from one tree to the next, supports entire ecosystems. I don't think we know enough about fungi yet. They are amazing. There's hundreds and hundreds of different species. A lot of them are saprophytic, meaning that they break down the stuff that's on the ground. So basically, the garbage, which of course in nature isn't garbage. Um, but yeah, they're very amazing. A lot of them are poisonous. The more pitiful they are, the more poisonous they usually are. Um, but a lot of them are not healthy. Um, but at the same time, they're also the source of a lot of our antibiotics, like your penicillin and that kind of thing. So they're pretty amazing organisms as they are. And of course, they also do, fungi, molds do a lot of damage to us in our environment, to our food, because they just want to live too. Of course, we also then utilize them again to make wine out of them or to bake bread, that kind of thing. Lots of diseases are made from are caused by fungi. um, this is a ringworm right here. um, and then we've got fungi are all over our body, whether as infections or just living on us, with us, around us at all times. It's a very normal thing, and a lot of them are opportunistic, which means that they'll get you when you're down, um, which is true in a lot of cases. And then of course we eat them too, right? And that's what I'm going to leave you with. Hopefully this was a little helpful.